Plant breeders and conservationists know that preserving the wealth of genetic diversity on Earth is key to securing our future. For a task this monumental, long-term storage solutions are essential, and that's where cryopreservation comes in. Cryopreservation is the storage of materials at ultra-low temperatures. Plant materials are cryopreserved in liquid nitrogen, which maintains a temperature of -196 degrees Celsius. That's colder than any environment where you'd find plants, yet their tissues can survive these temperatures for long periods of time with little degradation. After storage, these tissues can be warmed and brought back to the growing state. It's pretty amazing. There are methods available for cryopreserving seeds, pollen, cell cultures, dormant buds, and tiny shoot tips. Each of these fulfills a different role in our collection and must be treated appropriately for successful preservation and regrowth. When we want to conserve the genetic diversity found throughout plant populations, we typically preserve seeds, which are formed from the reproductive tissues. However, some plants don't make seeds. Some make seeds that cannot tolerate drying or cold temperatures. Some seeds just don't have the genetic makeup that we want. That's where dormant buds and shoot tips come in. These are derived from vegetative tissues that can be used to produce clones with the same genetic composition. This is important for preserving specific fruit cultivars that are propagated by grafting or cuttings. In dormant bud cryopreservation, branches containing buds are harvested from cold hardy plants during winter. They are then dried to remove any excess water that might form lethal ice crystals. After drying, they are slowly cooled to the point where the ice damage is no longer a risk, then plunged into liquid nitrogen vapors and stored in cryotanks. When they are needed, the dormant bud can be removed from the cryotanks, warmed, rehydrated, and then grafted onto rootstock. Alternatively, these buds may be sprouted and then placed into tissue cultures to regenerate clean plants. Dormant bud cryopreservation is preferred when plants come from cold hardy locations, established methods exist, and when infrastructure is available. This approach allows us to quickly get vegetative tissues into storage and maintain them for a long period of time. At the National Laboratory for Genetic Resources Preservation, we have cryopreserved apple buds that are still alive after more than 30 years. Another approach to cryopreserving clonal material is to isolate and preserve 1 mm shoot tips, which are the growing points in plant tissues. Shoot tips contain organized cell tissues called meristems that divide to make new cells. These meristematic cells are densely packed and will elongate and form large vacuoles that contain high concentrations of water and other substances when they mature. Actively growing shoot tips are well hydrated and can't be dried down like dormant buds. So we need to remove the water by immersing them in solutions called cryoprotectants. These chemical solutions dehydrate cells and can replace water with permeable chemicals that will help keep the cells alive during cryopreservation process. After cryoprotectant treatment, shoot tips can be safely plunged into liquid nitrogen. When it's time to recover them, the shoot tips are removed from the cryotanks and warmed. The cryoprotectants are diluted. Then the shoot tips are grown on a medium that provides nutrients and sugars to the plant. This method is very difficult and requires highly specialized facilities and skilled technicians. Additionally, clean plant material, tested methods for tissue culture, and proven cryopreservation methods are needed. While laborious, this method can be used for a wide range of plant species, including those with temperate or tropical origins. Dormant bud and shoot tip cryopreservation methods are essential technologies for preserving plant genetic resources on a long-term basis. This is why gene banks around the world have invested in necessary infrastructure and technical expertise to preserve collections in liquid nitrogen. Cryopreservation methods are also used by botanical gardens to preserve rare plants. Cryopreserved collections complement field collections, while field plantings or in situ reserves are readily available for research, propagation, and breeding. Collections stored in liquid nitrogen are protected against weather and pests and are cheaper to maintain on an annual basis. Clonal tissues are an important component of plant gene banking, and thanks to cryopreservation, we can maintain their secure backups to ensure the future of plant genetic diversity.